So today I'm going to be presenting Chapter 2, Segment 1, which is going to go from the specialized cells of the neurons all the way down to axons and axonal transport. So the first thing to note is that neurons are nerve cells and are the basic unit of the nervous system, and they're composed of the body, the dendrites, and the axon. There's about 89 to 90 billion in the adult brain, and they're meant to receive, integrate, and distribute information, and they're arranged in massive networks throughout your body. There's glial cells, which are non-neuronal brain cells that provide structural, nutritional, and other types of support for the brain. They're basically the cells that are going to supply neurons with the tools necessary to do their function. So neurons were first thought to be continuous with one another, forming endless networks without any spaces, and that was first proposed by the neuroscientist Camillo Golgi. This was then challenged by neuroscientist Santiago Ramon y Cajal, who proposed that neurons came very close to one another but were not continuous, that there were actually gaps between them. This was then put into words with the neuron doctrine, which hypothesized that the brain was composed of separate cells that are distinct structurally, metabolically, and functionally, and that the information was transmitted across these tiny gaps. This gap was called a synapse, and it's basically the tiny gap between neurons where the info is passed from one to another. And the first demonstration of these synapses were performed by Sir Charles Sherrington, and it's estimated there's about a quadrillion synapses within the body. The neuron also has 4 distinct structural divisions that are specialized for information processing. Within each neuron, there's going to be the mitochondrion, which is the cellular organelle that provides metabolic energy for the cell. The cell nucleus, which is the spherical central structure of the cell that contains DNA and chromosomes. And ribosomes, which helps with the structure of the cell body, where the genetic info is translated to produce proteins. There are 4 divisions of each neuron. There's the input zone, the integration, conduction, and output zone. The input zone is a part of the neuron that receives information from the other neurons or from specialized sensory structures. This usually correlates to the dendrites, which are the extensions of the cell body that are receptive surfaces of the neurons, branched to accommodate synapses. The dendrites are where the one neuron is basically receiving information from another neuron. The integration zone is a part of the neuron that initiates the nerve's electrical activity, and that corresponds to the nerve's axon hillock, and also, for most of them, the cell body, which is a region of the neuron where the nucleus is present. This area of the cell body also receives additional synaptic input. Next is the conduction zone, which is where the nerve's electrical signal may be actively propagated. And that is usually corresponding to the axons, which is a single extension from a nerve cell that carries action potentials from the cell body down to the axon terminals to other cells. There's also axon collaterals, a branch of an axon which comes from a single neuron. And the last part is the output zone, which is where the cell sends information to the next cell. And that corresponds with what I had said before, the axon terminals, which is the end of an axon or axon collateral that actually forms a synapse on another neuron or another target cell. So there's some classifications of neurons that can be done by shape, size, and function. There's 3 main types of neurons by shape. There's the multipolar cell, which is a nerve cell that has many dendrites in a single axon. So what you'd see is a cell body with multiple dendrites coming off it and one axon going straight down. There's a bipolar neuron, which is a nerve cell that has a single axon at the end, single axon at the other end. So you're going to have the dendrite, and then the axon, and then the axon terminals. And then there's a unipolar neuron, which is a nerve cell with a single branch that leaves the cell body, then extends in 2 directions. So one is the receptive pole, while the other is in the output zone. So in this case, the cell body is actually within the conduction zone of the neuron. So it's between the axons, the cell body, extending out. There's also classifications by function. So there's the motor neuron, which is a nerve cell that transmits motor messages by stimulating a muscle or gland. This one usually has long axons that reach out to synapses on the muscles, and they also cause contractions in response to commands. The sensory neuron, which is a neuron that is directly affected by changes in the environment, can be affected by light, odor, touch. It just carries messages from the periphery to the spinal cord and brain. And then lastly, there's the interneuron, which is neither a sensory nor a motor neuron. It receives information from other neurons, processes it, and then passes the integrated information to the other neurons. And they make up huge networks and circuits throughout the body. Information between neurons is received through synapses. So there's a few things that we just have to define before we can get into that. First is arborization, which is the elaborate branching of the dendrites of certain neurons. There's presynaptic, which refers to the region of a synapse that releases the neurotransmitters to the other neuron, and postsynaptic, which refers to the region of the synapse that receives and responds to the neurotransmitter. So a synapse generally has three principal components: the presynaptic membrane, which from the definition would be corresponding to the axon terminals of the neuron that transmits the information by releasing the neurotransmitters. There's a synaptic cleft, which is a space between the presynaptic and postsynaptic elements. And the third part is the postsynaptic membrane, which is the membrane on the surface of the cell that's receiving the information by responding to neurotransmitters from the presynaptic neuron. So there are also synaptic vesicles, which are small spherical structures that contain molecules of neurotransmitters. They're generally located on the presynaptic axon terminals, and they're just tiny hollow spheres that contain the molecules that bring them to the postsynaptic membrane. A neurotransmitter, by definition, is the chemical that's released from the presynaptic axon terminal that serves as a basis of communication between neurons. They're released when the synaptic vesicle ruptures due to electrical activity. So they'll diffuse across the cleft and interact with postsynaptic receptors and cause electrical changes in the postsynaptic cell. A receptor is a specialized protein molecule that captures and reacts to these neurotransmitters or hormones. Neurotransmitters generally don't enter the postsynaptic neuron, but they bind to these receptors momentarily and then dissociate. Most postsynaptic membranes have a high density of these receptors in order to keep quick movements between neuron to neuron. And there's also the dendrites on the other neuron that change shape in response to new patterns of synaptic activity and the formation of new neural networks. So this is called neuroplasticity, and that's the ability of the nervous system to change in response to experience or the environment. A good example of this would be dendritic spines, which increase the surface area of dendrites, allowing for extrasynaptic contacts. Another thing to do is try to visualize the cells of the brain, and this is done through many chemical and scientific processes. This is called histology, which is the study of the composition of tissue. So the most common traditional type of stain is a Nissl stain, and it outlines the cell bodies because the dyes are attracted to the RNA distributed within the cell. This type of stain allows us to measure the cell body, the size, and the density of cells in a particular region. The next one is the Golgi stain, which labels only a small minority of the neurons in a sample, but it stains them very deeply and completely, and allows us to see fine details of the cell structure like the dendrites and axons. And this will help us quantify the types and shapes of neurons that are present in a region. Another technique is audioradiography, which is a histological technique that shows the distribution of radioactive chemicals in tissue. Another would be immunohistochemistry, which is a technique that labels antibodies and are used to visualize the histological distribution of specific proteins. This is followed by in situ hybridization, which is a technique in which labels complementary nucleic probes that are used to identify cells expressing specific messenger RNA transcripts, and this reflects the activation of specific genes of interest. And lastly, they use tract tracers, which are compounds used to identify the routes and interconnections of neural projections. So now that we have a little bit of background on how we can see these, we can get on to how the axon integrates and transmits information. So the axon integrates in the integration zone through the axon hillock, which is a cone-shaped area from which the axon originates out of the cell body. This area gathers and integrates information from all the synapses of the neurons, dendrites, and soma, then converts and processes this information into code of electrical impulses to carry these messages down the axon to the target cell. This is a process of innervation that means to provide neural input, and it occurs through the axons and axon collaterals. Important substances that are needed in the axon terminals are transported by transport vesicles this way. Motor proteins are what transports these vesicles. Motor proteins are specialized kinetic protein molecules that convey a load, such as a vesicle, from one location to another. So they walk these cells through the axon to the target. And this is a process of axonal transport, which is transportation of materials from the neuronal cell body to distant regions in the dendrites and axons, and from the axon terminal back to the cell body. And there are two main types of axonal transport. There's anterograde transport, which moves matter toward the axonal terminal, and retrograde transport, which moves it back to the cell body for recycling. And that is the conclusion of the Segment 1, Chapter 2 outline.